Hello everyone, welcome and greetings of the day. I am going to introduce you today to identification of different herbal actives which are present in the formulation. In our earlier lessons we saw how these traditional preparations are prepared, but all these traditional preparations are therapeutically active only when they contain the right amount of phytochemicals, actives or bioactives. So, let us see today how can we detect the presence of these bioactives in the formulation or in the herb before using it in the formulation. Herbal medicines are used widely in Ayurveda. We looked at different traditional Ayurvedic preparations. The traditional Chinese medicine uses herbs, and the modern phytotherapy also has a lot of herbals now included in their system of medicines. The therapeutic activity is due to the secondary metabolites such as alkaloids, flavonoids, tannins, terpenoids and glycosides, and all these constituents are responsible for the therapeutic activity of the herbs. Phytochemical screening means we are going to screen this herb for presence of different kind of bioactives which are present. This is a systematic approach to detect and characterize different kind of constituents which are present in the herb. It serves as a foundation for pharmacognosy, drug discovery and herbal drug standardization. It also provides a scientific evidence for traditional claims, helps to identify potential lead compounds for new drug discovery and development, serves as chemotaxonomic markers to distinguish plant species. It ensures quality control and safety in herbal formulation, enables pharmacological correlation between the bioactive compound and therapeutic uses. Unless we identify whether this constituent is present in a herb, how do we ensure the therapeutic activity? Now, for us to detect the presence of this constituent in the herb, we first have to ensure a good concentration of this constituent in the solution that we are going to find out. So, a step for this would be extraction. It is a process of separating desired phytochemicals from the crude plant material using selective solvents. So, when we are including a herb in a formulation, it would be wiser to separate the constituents from the main herb matrix and then include it in the formulation. So, not only for identifying the actives, but for them to be included in the formulation, it is very important that we subject the herb material to extraction. This is the first and most critical step in phytochemical screening or in formulation. For example, extracting the alkaloids from cinchona bark. These alkaloids are known to be excellent antimalarials. So, these can be extracted with acidic solvents or the volatile oil which is extracted from clove buds using steam distillation. So, this is a simple method of extraction shown here. This is a source of heat, we have water and a steam is generated which is passed through this material which is kept with water or without water, and this steam then carries the volatile oil, and here we have a receiver separating water and the essential oil. This is generally used for all volatile oil containing materials. So, suppose you have the clove buds, we have lemongrass, we have eucalyptus, we have mint, any material that contains the volatile oil can be placed here where the steam carries the volatile oil with it, and here it is separated and collected. The objectives of extraction is to improve purity, stability and bioavailability of these herbal extracts, to remove the unwanted material and cell matrix, that is these constituents are present in a particular cell. So, we just are interested in the core of the cell, these constituents. So, to remove that, this process is called extraction, to obtain maximum yield of these bioactive compounds and to concentrate the phytochemicals for screening, isolation or for formulation. The conventional methods of extraction is maceration and percolation, and most of the indigenous system of medicines use only maceration or percolation as extraction methods, using natural solvents like water, milk, cow urine or ghee. Soxhlet extraction and reflux extraction are being added to modern day methods of extraction for separating the active constituents. The advanced extraction methods use ultrasonic assisted extraction, microwave assisted extraction and supercritical fluid extraction. This uses the ultrasonic waves, microwave assisted uses microwaves to enhance the penetration of solvent into the cell matrix. This is simple maceration where the drug is kept in contact with the solvent for 3 to 7 days, and periodical stirring assures that the solvent surrounding each drug particle is exposed to a new solvent around it. So, by just swirling this, whatever solutes have diffused in the solvents are just distributed evenly. This is then simply filtered and this is our macerate. So, this is just by keeping the drug in contact with the solvent. Percolation is the herb, this is a percolator. Herb is packed here and the solvent is added from top. The solvent percolates down this batch of herbs which is packed here. So, each time the herb is exposed to a new solvent that is percolating. As the solvent is going down, it carries the active constituents with it. So, whatever is collected below this perforated plate is the percolate which has our actives from the herb. So, what is left behind is the cell matrix or the plant matrix after extraction. Soxhlet is a very common lab scale method used to separate. Here the herb and the solvent is in contact with each other. With the heat, this mixture is heated, solvent vapors rise from here, through this tube they go into the condenser. This is the condenser. When these vapors are condensed, they are collected in this tube, in this thimble tube. So, when the level reaches the siphon tube, this is siphoned into the flask again. So, this process continues till we get a very dark extract here, but we should keep in mind that herbs which are not affected by heat can be extracted by Soxhlet apparatus. This is a continuous hot method of extraction. So, what happens is, every time the drug is packed here in the thimble, every time this drug is exposed to pure solvent, solvent here is rising, condensing and falling. A process of maceration and percolation also happens here. This is our drug packed. So, the drug is in contact with the solvent, a process of maceration is happening, and as the solvent goes, it is a process of percolation is also happening. So, this is considered to be continuous hot extraction method, best suited for drugs not destroyed by heat. reflux extraction is to prevent the solvent loss and continuous hot extraction, but here the solvent remains in touch with the same amount of drug. Ultrasonic assisted extraction uses ultrasonic waves to disrupt the cell walls and it improves the extraction efficiency. Microwaves are used for solvent penetration, and supercritical fluid extraction is where carbon dioxide is at a temperature and pressure where it behaves like a solvent and it behaves like a gas. So, like a solvent it penetrates very easily and it leaves because it behaves like a gas also. So, there is no solvent residue found in the extract. This is generally used for separation of caffeine from caffeine. That is how we have decaffeinated tea and coffee. Before we see what is present in the herb, we must understand what are primary metabolites and what are secondary metabolites. Primary metabolites are those compounds which are a direct product of photosynthesis. So, they are directly involved in normal growth, development and reproduction in plants, and they form the building blocks of life for the plants. These can be carbohydrates, proteins and lipids. So, this the plant uses for its day to day metabolism. Also, when a plumule is growing, it derives its nutrition from the stored carbohydrates, proteins and fats. So, all this is generally stored in the seed endosperm by the plant. Whatever extra primary metabolites are formed by the plants are converted to secondary metabolites using several enzymes and pathways, and these secondary metabolites are used by the plant for some other purposes like plant defense, adaptation and ecological interactions. They provide color, odor and bitterness that help to attract pollinators or repel the predators. For example, alkaloids which are produced as secondary metabolites from the primary metabolites are used for different pharmacological effects. Flavonoids are antioxidant and are used in UV protection. Tannins are antimicrobial agents and astringents. Terpenoids give flavors, and resins which are used for different therapeutic effects. Saponins, which have a detergent like properties, form a very important class of therapeutic activity for us. And some of the examples of the secondary metabolites are Taxol, which has become a modern day drug today, and the lead is a plant molecule. These are triterpenoids. Digoxin, which are glycosides and they are used as cardiotonics. These secondary metabolites are defense mechanism against herbivores, microbes and UV stress for the plants. It attracts pollinators because of the color, aroma and nectar. It contributes to flavor, fragrance and color in the herbs, again to attract pollinators, but for us when we use, they exhibit different pharmacological activities. This is the way secondary metabolites are classified, and here I have classified some of the commonly used secondary metabolites. Alkaloids, which are nitrogen containing compounds, example morphine and atropine. Glycoside, which has a sugar attached to a non-sugar part. Flavonoid, which has a benz-pyrrone ring and they are polyphenolic pigments. Tannins and phenols are again phenolic compounds. Terpenoids and steroids, this has a steroidal ring, the cyclopentanoperhydrophenanthrene ring present. Saponins again are a kind of glycosides, and coumarins. Factors affecting extraction: plant related, depending on the age of the plant, the moisture content in the plant, it affects the yield. Process related, when we powder it, what is the particle size. So, suppose you extract the herb as it is and you powder it and extract, it gives an increase in the yield. Use of the kind of solvent also decides how much extractive yield we are getting. Environmental conditions like temperature, time, agitation and the pH also defines the extractive yield. The drug to solvent ratio will help us decide an optimum yield. Generally, we keep the proportion of 1:5, but a higher proportion may be used for optimum extraction. To decide whether to use a polar or a non-polar solvent depends on the kind of constituents we want to extract. Suppose we want to extract oils, fats, waxes, chlorophyll and some terpenoids, we have to use non-polar solvents. However, the traditional systems of medicine like Ayurveda uses only natural solvents like water, urine, cow urine, ghee and some oils. So, whenever we have the lipophilic compounds and we are trying to make a traditional system of medicine, we use only the natural solvents for extraction. These may be used in modern day extraction for defatting the plant material, so that it becomes easy for us to extract the bioactive which is present inside. Moderately polar solvents like chloroform, ethyl acetate and ethanol can be used for alkaloids, terpenoids, flavonoids and steroids. Polar solvents like methanol and water can be used for polar constituents like tannins, glycosides, saponins, phenolics, flavonoid glycosides and polysaccharides. When we want to extract different constituents present in the same plant material, we can go for sequential extraction, where solvents are used step by step for the same plant material in increasing polarity. So, for example, we have a plant material, the solvents in the mentioned order, you first add hexane, extract, you separate the hexane fraction, keep it aside. To the same plant material, you add chloroform, keep the chloroform fraction aside. Then you add ethyl acetate, keep the ethyl acetate fraction aside. You add ethanol, separate the ethanol fraction, and finally you add water and retain this water fraction. So, we started this plant material by adding hexane. So, each time we added a solvent and separated, each solvent has separated different plant constituents from the same material. This is called fractional or sequential extraction. The benefit here is we can isolate different secondary metabolites each time we add a solvent. This allows selective recovery of compounds based on polarity. Non-polar solvents when used remove the fats, oils and waxes. So, hexane when we use removes all this. Moderately polar solvents like ethyl acetate is going to isolate alkaloids, terpenoids, flavonoids, and polar solvents when used will extract tannins, glycosides, saponins and phenolics. So, when we have a plant material, when we want to check the presence of all the constituents, then we can have this sequential extraction. This ensures maximum phytochemical diversity in the same plant sample. Coming to screening, the preliminary testing of plant extracts to identify the major classes of secondary metabolites. So, it can be qualitative and it can be quantitative. There are some methods where we can find out how much amount is present, but those are instrumental methods of quantifying the extracts where we use HPLC, GC. But today we will see qualitative methods of how we can detect the presence. Once we detect the presence, then we can go for quantitative measurements. Let us see how do we detect the presence of alkaloids present. These are nitrogen containing basic compounds. Example is morphine, where our alkaloids are present in the latex of this capsule. The preliminary test to detect, these are the general tests for alkaloids, and Dragendorf reagent, Mayer's reagent, Wagner's reagent, which contain iodine salts when added to a plant extract, and if they give an orange red precipitate, it indicates the presence of alkaloids. A cream colored precipitate with Mayer's reagent helps us detect the presence of alkaloids. Wagner's reagent gives us reddish brown PPT, and Hagener's reagent, which contains picric acid, gives us yellow crystalline precipitate, which confirms the presence of alkaloids. However, these are just preliminary tests and will tell us the presence of true alkaloids. Alkaloids again have different classes, and these will just give us a first idea whether alkaloids are present. Depending on the class of alkaloids, we have different tests. For example, the tropane class of alkaloids gives the vitally morin test positive. Caffeine alkaloids or purine alkaloids answer the muroxide test. So, depending on each class later, we have different class of alkaloids. The alkaloids from cinchona bark answer a confirmatory test called thalioquine test, but here we will just restrict to what are the preliminary tests which are used to detect these alkaloids. Glycoside, which contains a sugar and a non-sugar part. Now again, glycosides are classified as cardiac and anthraquinone. So, to detect the sugar, any test for sugar is done, like Molish test or Fehling solution will tell us whether a glycoside is present, but that test will detect only the presence of sugar. To detect the presence of the glycon present, the glycon tells us the class of glycosides. So, for cardiac glycosides, where there is a steroid ring present with a lactone member ring attached to this, example is digitalis leaf, this is digitalis purpurea, and anthraquinone glycosides, which we see in aloe and senna. Anthraquinones, these are quinone structures which are seen. So, for cardiac glycoside, Keller-Kilani test, which gives a reddish brown ring at the junction when ammonia is added to that extract. Legal's test, where there is pink to red coloration on addition of sodium nitroprusside. Anthraquinone test, the Bontrager's test is a very famous test to detect the presence of anthraquinone, which gives a pink or red coloration in the alkaline layer, and the free anthraquinones are detected under UV, which give bright fluorescence. Flavonoids, which are an important class of compounds, these are polyphenolic compounds responsible for pigmentation, antioxidant and therapeutic activities, and they are present in licorice. Quercetin is an important flavonoid. A very important test called Shinoda test is used to detect flavonoids. When magnesium ribbon and HCl is added, it gives pink to red coloration. This test helps us find out the presence of flavonoids. An alkaline reagent where the yellow color when added disappears after adding an acid. This also tells us the presence of flavonoids. A confirmatory test can be done on TLC by developing a fingerprinting and finding out the UV spectrum for flavonoids. Tannins and phenols can be detected with ferric chloride. When we add aqueous or alcoholic ferric chloride solution, there is a black blue coloration, and we can also distinguish whether there are hydrolyzable tannins or condensed tannins. Hydrolyzable tannins are elago or gallotannins, and condensed tannin, an example is catechin. Lead acetate test, where when lead acetate is added, it precipitates. This also helps us detect tannins. These can be again confirmed with TLC. So, for example, when we are trying to screen Terminalia arjuna, This is a cardioprotective herb and has cardiac glycosides, it has tannins, flavonoids and glycosides. So, all these three can be detected with the help of the phytochemical test which we just mentioned. Polyherbal extracts of Triphala, where all the three fruits contain tannins, by simple addition of ferric chloride the presence of tannins can be detected. The test for saponin glycosides can be conducted by simply taking the dry powder in a test tube, add water and shake it vigorously. There is a persistent froth which remains in the test tube. So, this is like when you add little soap powder to a test tube and shake it, we have the white froth formation staying persistently for 1 minute. That is the froth test for saponins. That is why they are called soap like saponins. Hemolysis is when you take the plant extract and add a drop of blood, the plant extract, if it contains saponin, leads to hemolysis of the red blood cells. This is the hemolysis test for confirming the presence of saponins. Steroids and triterpenoids can be determined with the help of Salkovsky's test, which gives a reddish brown color, and Lieberman-Burchard test. Lieberman-Burchard test uses acetic anhydride, which is now banned. So, performing this test may be limitation. However, we can perform Salkovsky's test to detect the presence of steroids. Coumarins, these are a class of flavonoids which contain benzpyrrone derivatives, and the UV fluorescence, this is the UV fluorescence seen here, a bright blue under the UV light will help us detect the presence of coumarins. Resins, again there are different classes of resins. For example, this is asafoetida, our normal hing. This gives fluorescence because of presence of umbelliferic acid. When acetone is added, it causes turbidity because these resins are not soluble in any polar solvent. So, when you add alcohol, it precipitates because of less solubility. When you add water to this, it forms an emulsion, that is because of an volatile oil and gum which is found in the resin. To detect volatile oils, when Sudan 3 is added, it causes a red coloration. So, wherever you see the oil droplets, these oil droplets take up the stain. So, this red coloration will help us detect the presence of volatile oil, and the sodium hydroxide soap like white precipitation is formed when the volatile oil is present. These are of course confirmatory tests using state of the art instruments to detect the presence of oil components. So, to summarize, to detect the presence of alkaloids, Dragendorf, Mayer and Wagner's reagents can be used. Cardiac glycosides can be detected with the help of Keller-Kelani's test. Anthraquinones, the best test to detect is Bontrager's test and modified Bontrager's test, where extra ferric chloride is used for hydrolysis. Flavonoids are detected by using magnesium ribbon and hydrochloric acid in case of Shinoda and alkaline reagent test. Tannins and phenolics can be used detecting ferric chloride and lead acetate. Saponins are detected by froth and hemolysis test. Steroids with Salkovsky and Lieberman-Burchard test, coumarins by UV fluorescence and NaOH, resins with acetone and alcohol, and individual resin tests are different. Asafoetida also shows a bright fluorescence under UV, volatile oils with Sudan 3 test and the NaOH test. Importance of these qualitative tests: it allows rapid identification of phytochemical classes in crude extracts, It is essential for routine quality control and herbal standardization, helps us detect adulteration, substitution or contamination, and provides a guideline for further pharmacological testing. Very important before we go for any costly analytical procedures of isolation. The WHO also recommends phytochemical screening as part of the herbal quality control. This ensures reproducibility and consistency of herbal products. It detects the substitution adulterations and provides a baseline for chemoprofiling. Finds a large application in drug discovery where bioactive compounds are detected from plant compounds. For example, detection of quinine for antimalarial activity, morphine for analgesic activity, and paclitaxel from the taxus tree for anti-cancer activity. This preliminary screening helps us reduce time and cost and gives us promising targets. It identifies herbs which are rich in antioxidant and bioactive compounds and supports the development of nutraceuticals and many important lead compounds for modern drug discovery. It serves as basis for bioassay guided fractionation, helps us isolate new compounds from complex herbal mixtures, provides chemical and pharmacological data to correlate activity and constituents, and has a rational selection of herb use. It ensures compatibility and synergy. For example, anti-diabetic capsules, saponin rich herbs, flavonoids which contain antioxidant activity are being developed as nutraceuticals. So, to summarize, this is an important gateway for in a drug discovery process. The solvent polarity and extraction method determine which phytochemical is to be detected or extracted, and these qualitative tests provide preliminary chemical profiles which of course require further confirmation, but gives us a very important basic idea. To conclude, the phytochemical screening bridges traditional knowledge and modern scientific research. It plays a vital role in quality control, standardization and drug discovery, enables evidence based use of herbal medicine and supports further herbal research, a foundational step in natural product research. Thank you.